Hello students, I welcome you to EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, Karnal in Haryana, India. Dear students, today I will talk about the evolution of enzymes so as to identify significant residues which can be useful for enhancing the performance of the enzymes, and also I will undertake gene duplications for creating new enzyme activities. We have seen in the last module that during evolution certain residue positions are conserved for either contiguous motifs or single identities. In addition, we have also seen that there are certain strong similarities as well as weak similarities in positions of multiple sequence alignment. All enzymes are proteins, therefore, we will necessarily be learning the evolution of proteins today. Proteins are linear sequences of amino acids encoded in the genes. The genes, after replication, are passed on to the next generation offspring. However, during replication, gene sequence may undergo point mutation either in its regulatory sequence or within the coding sequence of the protein. When it happens in the regulatory sequence, it will affect the concentration of the enzyme within the cell. When it occurs within the coding sequence of the protein, then it may affect the activity involving affinity or turnover number of the enzymes. Those individuals which acquire mutations suitable to survive in the changing environment will be selected as fittest. That is what we know, the well-known theory of Sir Charles Darwin, that is the survival of the fittest. In module 13 and 14, we identified active site residues of OSBS, a synthase, from its three-dimensional structure, having bound substrate and product. Because 3D structure of the HPRKP enzyme bound to its substrate or product, that is ATP or pyrophosphate, is not available. Therefore, in the last module, we identify its active site residues from the multiple sequence alignment involving both closely and distantly related HPRKP sequences. This shows that both the structure and the sequence approaches can be used to identify the residues which are significant for its catalysis. Therefore, these identified positions may be chosen for enhancing the performance of an enzyme in the laboratory. In addition to its main activity or major activity, sometimes an enzyme has some minor activity, side activity or side effect activity, which we usually call promiscuous activity. So, during replication, we can also have, in addition to point mutations, sometimes complete gene duplication. So, it may happen that complete gene is duplicated to create another new copy, and then subsequently the new copy may accumulate point mutations to convert this minor activity of the enzyme to some main activity, so that thereby resulting in the evolution of a new enzyme in this duplicated copy. In this module, we will combine the consensus primary sequence information with the consensus secondary structure to identify point mutations for improving the performance of the enzyme, as well as we look at gene duplications for acquiring new enzyme activity. Thank you. Therefore, the learning objectives for understanding evolution of enzymes include compiling a set of orthologous enzymes with known kinetic values such as Km and Kcat. Then we need to predict consensus sequence of the enzyme family, which determines the kinetic parameters of the enzyme. We will also predict the consensus secondary structure, which is mapped onto the predicted consensus sequence for correct folding of the enzyme. This can be achieved with multiple sequence alignment application available at Promels 3D. Then we'll map the residues which are significant for improving performance of the enzymes. And finally, we'll look at gene duplication for creating paralogs with new activities or subunit interactions for cooperativity. Dear students, orthosuccinyl benzoate synthase, that is OSBS, belongs to the enolase superfamily of the enzymes and is characterized by the presence of an enolate intermediate which is generated by the abstraction of the alpha proton of the carboxylate substrate. It catalyzes the dehydration reaction to produce orthosuccinyl benzoate. In the modules 13 and 14, we took the example of this enzyme, that is OSBS, to search PDB database and found that enzyme structures bound to its substrate as well as product were present there and were used to detect the active site residues from these complexes. In this module, we will carry forward the same example to identify other essential residues for enhancing the performance of the enzyme. Dear students, Km of an enzyme reflects its affinity for the substrate, and low Km enables enzymes to work at low substrate concentration. During enzyme engineering, we may be interested in decreasing the Km of an enzyme so as to enable it to work at low substrate concentration, that is to increase the affinity. Similarly, Vmax of an enzyme, that is the maximum velocity, which is the product of the kcat, that is the turnover number, and the total enzyme concentration present, reflects its catalytic or turnover number, so that an enzyme with high turnover number is able to perform better at lower concentration of the enzyme. So, in both the situation, either at low concentration of substrate, that is low Km, or high turnover number, that is low enzyme concentration, enzyme industry may be interested in altering any of these two values, either Km or the turnover number, so that when we increase the turnover number of an enzyme through enzyme engineering techniques, so that the concentration of the enzyme which is required for the reaction is low. On the other hand, catalytic efficiency of an enzyme is measured by the ratio of kcat, that is turnover number, to Km, that is affinity. Therefore, enzyme efficiency can be increased by either increasing the kcat or lowering the Km value. Highest catalytic efficiency is defined as that as soon as the substrate diffuses into active site and passes out after its conversion to the product. That is, this is transformed to the product without consuming further time, I mean it diffuses and goes out, and during this passing the substrate is converted into the product. Consequently, an enzyme may achieve its efficiency up to the upper limit of the diffusion of the substrate to the active site, which is in between 10 raised to the power 8 to 10 raised to the power 9 per mole per second. This is exemplified by highly evolved enzymes such as acetylcholine esterase, triose phosphate isomerase, carbonic anhydrase, and some others also. However, majority of the enzymes has achieved catalytic efficiency approximately 1 million per mole per second, with normal distribution from almost 1 to approximately 10 raised to the power 8 to 10 raised to the power 9, and the peak being at 1 million, that is 10 raised to the power 6 per mole per second. The OSBS is having its catalytic efficiency to approximately 1 million, and it has 80 members, including both closely related and distantly related or divergent amino acid sequences, as well as it has also 4 3D structures. The OSBS from E. coli has its catalytic efficiency around 2 into 10 to the power 6, that is 2 million per mole per second. And on the other hand, an OSBS, that is same synthase enzyme from Thermobifida fusca has catalytic efficiency less than 1 million at the level of 0.67 million per mole per second. Therefore, OSBS with catalytic efficiency on either side of the normal population with 1 million per mole per second and having 80 closely as well as distantly related members. Therefore, OSBS is an excellent model system for elucidating the principles of enzyme performance so as to understand to be used in further enzyme engineering. But before identifying the amino acid residues affecting the performance of OSBS, let us check if kcat and km data values are available for mutated OSBS analogs. Therefore, visit enzyme database at XPASI and search for orthosuccinyl benzoate synthase. This search displays enzyme 4.2.1.113 for orthosuccinyl benzoate synthase with several alternative names. Now visit Brenda enzyme kinetics database and search for the EC number 4.2.1.113 and press Start Search button. The EC number 4.2.1.113 is the required enzyme and is returned. Therefore, click this enzyme EC number and in the subsequent page, limit your search to E. coli and expand its functional parameters. KM data from E. coli reveals that there are several mutations which result in both increase and decrease in KM value. That is, the affinity of the substrate for OSBS is increased or decreased by these mutations. However, most of the mutations leads to increase in KM value, that is reduced affinity from 0.012 millimolar for wild type enzyme to higher numbers. This can be understood simply because these mutations were planned in the laboratory and the mutated OSBS is not subjected to environmental pressure for the selection of the fit test, for the survival of the fit test enzyme with highest affinity. But nevertheless, this data will help us understanding point mutations for altering affinity and consequently substrate specificity. For example, mutation at serine 262 which is detected in the binding site of enzyme substrate 3D complex result in increased Km. Mutation to threonine result in drastic increase in Km as opposed to slight increase in mutation to glycine. The mutation at 265 from isoleucine to alanine increases the affinity by a factor of 10, and therefore this data will be very useful in understanding the mutations which lead to the alteration in the affinity of the enzyme for its substrate. Similarly, kcat data from E. coli reveals that there are several mutations which result in both increase and decrease in kcat, that is the turnover number of the synthase for its substrate. There are several mutations which leads to increase in kcat. This data will help us understanding the point mutations for altering the turnover number and consequently the Vmax. In addition, different mutations at the same position may have different effects on the turnover number. For example, mutation at serine 262 glycine results in decreased kcat, whereas mutation to threonine enhances the turnover number. In addition, kcat over km, that is kcat to km ratio list for E. coli reveals that wild type enzyme has catalytic efficiency, that is Kcat/Km ratio at 2000 millimole per second, millimole per second, that is 2 million per mole per second, and the substitution of serine at 262 with glycine reduces the catalytic efficiency by 1/4, whereas the substitution with threonine at the same position results in reduction of the catalytic efficiency, that is Kcat to Km, to 1/10. This clearly reveals the involvement of serine 262 in the binding of the substrate, as the catalytic efficiency is not affected drastically, say by a factor of 1000 or like that. In addition, the Kcat/Km, that is ratio list for E. coli, reveals that the wild type enzyme is mutated twice at position 51. In addition, mutant at position 51 has another mutation at position 48. The position 48 was also mutated individually to see the effect of mutation at position 48 only. The substitution of phenylalanine at 51 with alanine reduces the efficiency to 400 from 2000 per millimole per second, whereas the substitution with tyrosine at the same position results in reduction to 350. However, second mutation at 48 from leucine to methionine in already mutant at 51st position for phenylalanine to tyrosine increases efficiency to 370, whereas single mutation at 48 from leucine to methionine with results in 570, which is better than even double mutant with catalytic efficiency of 370. From these results, it is clear that all mutations reduced catalytic efficiency of the wild type from 2000 per millimole per second to lower numbers. These catalytic efficiencies reveals that all mutant enzymes has resulted in unfavorable catalytic efficiency. The periodic spectrum of amino acids shows that mutations at position 48, 51, 262, 288 were planned as per physicochemical properties. For example, replacement of leucine with methionine at 48, or phenylalanine with tyrosine at 51, or serine with threonine at 262, or glycine with alanine at 288 were introduced, keeping physicochemical properties in the periodic spectrum of amino acids. Therefore, mutating any of these residues as per physicochemical similarities is expected to increase or decrease catalytic efficiency, but slightly, not drastically. However, drastic reduction in Kcat/Km from 2000 to 3.8 indicates that only physicochemical criteria for planning mutations is not sufficient. Therefore, let us check if the secondary structure at these positions is also important for evolving catalytic efficiency of the enzymes. For this purpose, we need consensus sequence for each member, orthologous member of the family, as well as consensus secondary structure, which can be predicted using multiple sequence alignment program PROMALS 3D. For constructing multiple sequence alignment, we need, however, closely as well as distantly related OSBS amino acid sequences. Visit UniProt knowledge base and search for orthosuccinyl benzoate synthase. Limit the search to reviewed 82 and select 250 from show drop down list to display all 82 sequences in a single page. Now sort the entries on the basis of length. The first two entries with unusual length of 1715 and 451 do not belong to orthosuccinyl benzoate synthase. The next entries with length 371 to last entry with length 320 are all of orthosuccinyl benzoate synthase. Therefore, limit the search to length from 320 to 371 by clicking on the restrict range of length and enter this range in the search text box, that is 320 to 371. Now select all and download 80 selected sequences in uncompressed format. The entries will appear in the browser window. Copy on the clipboard and paste in the notepad. Find the entry with identification P29208. This is an entry for E. coli. Copy it and paste it at the end of the file. Save the file with a file name having extension .fasta and save as type all files. Promels 3D is a profile-based multiple sequence alignment application which combines Cypred to predict secondary structure for constructing multiple sequence alignment. Promels 3D first aligns similar sequences and then uses Cyblast and Cypred results for aligning divergent sequences. Promels 3D applies more elaborate techniques to align relatively divergent clusters. It allows extraction of sequences from the PDB structure files also. Therefore, upload the saved sequences in the file for 80 OSBS sequences in FASTA format and also upload one FHU OSBS structure and click submit button. This will begin multiple sequence alignment and will update the progress every 30 seconds. When the alignment is complete, display colored alignment in the order of input sequences with 60 residues in each row and displaying residues with consensus level at 0.8. Therefore, in each column of the consensus for the residues to the level of 0.8, that is 80% will be evaluated before displaying the residue in the consensus category either as identity or some other category which we will discuss later. The multiple sequence alignment with Promels 3D returns the consensus amino acid sequence in the second last line of the alignment block of the 60 residues for all the sequences and the consensus predicted secondary structure in the last line for this block. The secondary structure elements are represented with symbol lowercase h for alpha helices and symbol lowercase e for extended beta strands. The positions without any of these symbols are either turns or loops. Identical amino acids, that is with the chosen consensus level 0.8 in the present example, are displayed, that is the identical amino acids are displayed in uppercase and bold letters. The other consensus amino acid positions are categorized under 11 categories which we will look later on. The absolute conservation within or at the ends of secondary structure revealed the presence of catalytic residues. We know from the modules 13 and 14 that lysine at position 133 is a catalytic base for abstraction of the alpha proton from the carboxylate substrate. Similarly, aspartate at 161, glutamate at 190 and aspartate at 213 are involved in the coordination of the magnesium ion to stabilize alpha carboxyl group in the transition state. The arginine 235 is involved in the stabilization of enolate ion position in the transition state. Since these 5 residues are involved in catalysis, that is the catalytic mechanism, mutations at these positions will abolish the activity completely. The Promels 3D found these 5 residues conserved. Therefore, if a mutation at any of these conserved residues abolishes the activity completely, that means the residue is actually involved in chemical mechanism. Consequently, Promels 3D MSA can be used for identification of catalytic residues in the enzyme for which catalytic residues are not yet identified. So, the identical positions mean conserved position if leads to abolition of activity mean those identities or conserved positions are actually involved in chemical catalysis. Dear students, in OSBS, the lysine 133, the lysine 235 and aspartate 161, glutamate 190, aspartate 213 are the residues involved in the chemical catalysis. Therefore, mutating any of these catalytic residues is expected to abolish the enzyme activity completely. The mapped active site for OSBS shows that a magnesium ion is coordinated by aspartate 161, glutamate 190 and aspartate 230 from, let us say, top, and one oxygen from the substrate also coordinates to this magnesium ion. The literature information shows that lysine 133 comes from one side and lysine 235 comes from the other, and they are positioned to function as lysine 133 is to abstract the proton and lysine 233 to stabilize the transition state with the help of magnesium ion. Therefore, in this acid-base catalysis, the dehydration reaction of the substrate to produce the OSB product occurs. This is simply, you can believe that this is the substrate which is having carboxylate group. This is the magnesium which will be having coordinated with three acidic residues, that is aspartate, glutamate and aspartate. This magnesium then have the fourth coordination position from the carboxylate group of the substrate. From this side, the lysine 133 abstracts a proton so that a negative charge, that is an enolate anion, is formed, and this then goes to form a double bond, wherein the double bond in the carboxyl group breaks, and then the lysine 235 helps in breaking that, and therefore the charge is distributed here. In this way, the transition state is stabilized, and then the lysine 235 then converts back the transition state, and then the electrons returns and hydroxyl group from this side goes away with the help of the water molecule or having a hydrogen ion bound to it, that is hydronium ion, and water is removed and dehydration takes place. So, in this way, all these 5 residues with the help of magnesium ion helps to catalyze the reaction. The main function here is the abstraction of the proton so that a negative charge develops, which is stabilized by the magnesium and the lysine present on the other side. The MSA found serine 262 conserved at the end of a beta strand at position 262. Therefore, mutation with threonine or glycine result in decrease in catalytic efficiency of the OSBS synthase. However, surprisingly, mutation with threonine, although a strong similarity, but still it results in 10-fold decrease in catalytic efficiency, whereas replacement with similar tiny amino acid glycine at the same position at the end of the secondary structure still allows some comfort with reducing efficiency by a factor of 4 only to 480. This shows the absolute requirement for conserved arginines as both increasing size with preserving hydroxyl or decreasing size without hydroxyl in glycine reduce the catalytic efficiency. However, the reduction in catalytic efficiency just by a factor of 10 or 4 involves the involvement of the position in binding rather than in catalysis. Therefore, Promal's 3D MSA can be used for identification of binding residues in enzymes for which the binding residues are not yet identified. The bound product in the 3D complex with synthase reveals the presence of serine 262 inside the binding pocket for positioning the substrate. Therefore, mutating serine 262 is expected to affect catalytic efficiency, but not drastically, as serine 262 is not involved in catalysis. The 3D structure of OSBS bound to product OSB shows presence of hydroxyl group of serine near to the carboxylate group of succinyl moiety of the product OSB. Therefore, this helps in increasing the affinity of the enzyme for the substrate or the product. For mutating a residue in its 3D structure in PDB file opened with Swiss PDB viewer, we have to use the commands which are there in the toolbar. Therefore, run Swiss PDB viewer and open PDB structure 1FHV containing OSBS bound to its product OSB. Display the residue to mutate, that is serine 262 in the present case, and select mutate tool, which is the second last tool in the toolbar. It will ask to pick a group in the context menu. Choose serine 262 by clicking over its alpha carbon, and from the ensuing context menu choose threonine for mutating to threonine and click mutate tool again and respond by accepting mutation serine 262 to threonine 262. This will mutate the residue to threonine and will display the threonine in place of the serine. Replacing serine with a threonine preserves its hydroxyl group for affinity, which is shown here with hydrogen bond displayed in dotted green line on the left side of the slide, but introduces a methyl group which clashes with the carboxylate group of the succinyl moiety of the OSB, which is shown with pink color clash line from carboxylate of OSB to methyl group in threonine on the right side of the slide. Thereby, this reduces the catalytic efficiency. So, introduction of this clash is actually responsible for reducing the catalytic efficiency. Therefore, predicted conservation at 262 indicates that this position is significant and shall not be mutated for improving the enzyme efficiency. The mutation with glycine leaves an open space to reduce catalytic efficiency. Therefore, predicted conservation at 262 indicates that this position is significant and shall not be mutated for improving enzyme catalytic efficiency. The conserved residues glycine 126, proline 154 and glycine 240 are present within short stretches. These conserved glycine and proline within short stretches are required for introducing a turn or small loop to change the direction of the secondary structure advancement for correct 3D folding of the protein. Therefore, predicted conservation at 126, 154 and 240 indicates that these positions are significant and shall not be mutated, as it will deteriorate catalytic efficiency of the synthase. The predictions for the presence of glycine 126, proline 154 and glycine 240 within short stretches revealed introduction of turn or small loop in 3D structure, and it was actually found in the available 3D structure of E. coli OSBS as displayed in red colored turns and loops in this slide. A conserved glycine 288 is present at almost within a loop of 11 residues. Available kinetic data for mutations at this position reveals drastic reduction in kinetic efficiency from 2000 to 3.8 per millimole per second. The drastic reduction in kinetic efficiency for mutations at this conserved position is expected. Let us have a look at the reason for the drastic reduction in the kinetic efficiency of mutation at this conserved position within a long loop. Replacement of glycine with alanine at position 288 reduces the catalytic efficiency by a factor of approximately 500. From 3D structure, it is clear that mutation from glycine to alanine at position 288 results in the clash of the introduced methyl group in alanine with carboxyl moiety of the succinyl group of the substrate. Therefore, substrate is not able to fit or orient itself in the binding site correctly, thereby reducing the catalytic efficiency. Let us have a look at the presence of conserved residues within long secondary structures. Conserved arginine at position 27 is present at almost within the middle of a long beta strand of 14 residues, and another conserved residue, that is arginine at 159, is present exactly at the middle of a beta strand of 5 residues. Available kinetic data for mutation at position 159 reveals drastic reduction in the kinetic efficiency from 2000 to 11 per millimole per second. The drastic reduction in kinetic efficiency of mutations at this conserved position is expected. Let us now have a look at the reason for the drastic reduction in kinetic efficiency of mutations at this conserved position within a long secondary structure. A conserved arginine and 159 present exactly within conserved pattern of hydrophobicity with spacing of 2 having intervening variable hydrophilic residues. This spacing of 2 with variable hydrophilic residues suggests a beta strand on the substrate, beta strand on the surface of the enzyme. The conservation of arginine in beta strand on the surface indicates its interaction for stabilization of the 3D structure through electrostatic interactions involving the long arm in the arginine. Therefore, replacement of arginine with methionine in E. coli reduces its catalytic efficiency by a factor of 200. This suggests the involvement of arginine in folding. The presence of arginine in E. coli OSBS at position 159 reveals its electrostatic interactions with the main chain carbonyl from isoleucine 286, connecting two regular secondary structures separated by more than 100 amino acids in the primary sequence. We do not have the kinetic data for mutation at conserved arginine at position 27 present at almost within middle of a long beta strand of 14 residues. Therefore, let us see if this arginine is involved in electrostatic interactions for connecting two regular secondary structures separated by a long stretch of amino acids in the primary sequence. In Swiss PDB viewer, choose arginine 27, then choose compute hydrogen bonds command from the tool menu, then from the display menu, choose show only hydrogen bonds from selection, and then from the display menu again choose show only groups with visible hydrogen bond and display complete chain as ribbons from 5th column of the control panel. It is clear from 3D structure that conserved arginine at position 27 has several electrostatic interactions with carbonyl groups of the main atoms of distantly placed serine 264, isoleucine 265, serine 46 and proline 47. Therefore, mutating this residue will result in decrease in catalytic efficiency of the enzyme. In this slide, all aligned positions are displayed. Identical amino acids with chosen consensus level, that is 0.8 in the present example, are displayed in consensus sequence in uppercase and bold letters. For example, for the first 60 aligned positions, we have tyrosine, proline, arginine, then 2 glycines followed by a glutamate as identically conserved position. The non-identical consensus amino acid positions are categorized under 11 categories by Prommel's 3D and displayed using separate symbols. The presence of hydroxyl group containing serine or threonine is indicated by lowercase o letter. The negatively charged residues aspartate and glutamate is indicated by negative sign, minus sign, and positively charged residues lysine, arginine or histidine is indicated by a plus sign. The lowercase l in italics indicate presence of any of the three small aliphatic amino acids, valine, leucine or isoleucine. The conservation of any of the four tiny amino acids, glycine, alanine, serine or cysteine is indicated by lowercase t. Sometimes more liberal mutations are allowed at particular positions. For example, any one of the 9 polar amino acids, that is aspartate, glutamate, histidine, lysine, asparagine, glutamine, arginine, serine, threonine present in an aligned column is indicated with the lowercase p letter for the polar residues. This group adds 4 residues, that is asparagine, glutamine, serine and threonine to 5 charged amino acid groups indicated with lowercase c. There may be any one of the 9 small amino acids indicated with the lowercase letter s. The conservation of any of the 5 charged amino acids in a column is indicated with lowercase c letter. Any one of the 10 bulky residues is indicated with lowercase b, and any one of the 11 hydrophobic residues is indicated with the lowercase h. The @ symbol indicate the presence of any one of the 4 aromatic amino acids, phenylalanine, tryptophan, tyrosine or histidine. These categories indicate the reservation of the one of the 11 types of the amino acid groups in a particular column, which actually represent the requirement of the type of residue at the corresponding 3D position in the structure. Therefore, positions with the residues, especially for the first 5 groups, falling within loops indicate mutations for improving enzyme performance. For example, the mutation at position 265 from isoleucine to alanine increases the affinity for the substrate by a factor of 10, and this is a mutation categorized under group 4 for the aliphatic residues. Dear students, till now we have seen that multiple sequence alignment consensus sequence along with consensus secondary sequence or secondary structure provides insights into detecting significant functional residues in the enzymes. Now, let us have a look at another aspect of enzyme evolution, the complete gene duplication, that is one gene during replication duplicated and creating the additional copy of the same gene. So, we have the original gene and a copy. 2 situations at this stage may arise. First is, if the original gene has some other activity that will be also present in this duplicated copy. Let us say this is minor activity, also known as promiscuous activity. Then this additional copy may undergo point mutations to convert this minor promiscuous activity into the main activity, and thereby creating a new enzyme which will add enhanced capability to the individual so that it can now perform better and survive better. We know that enzyme OSBS has minor epimerase activity. We will take example of OSBS, that is synthase with promiscuous epimerase activity for gene duplication, and then we will analyze the point mutations to evolve this promiscuous activity into the separate epimerase activity, thereby enhancing capability by adding a new enzyme in the individual. Secondly, a gene duplication may enhance performance of an individual by evolving cooperativity among the subunits as exemplified by hemoglobin. Let us have a look at the case of the hemoglobin. Originally, let us say there was an individual or an organism species with one globin copy. This globin copy will transport the oxygen. This is the main function. This is the main function of the globin. Let us say that there is gene duplication creating the second globin copy. We will call them hypothetically as alpha or first hemoglobin or alpha hemoglobin and beta globin. In this way, now the organism or the individual has two copies, so its capability to transport oxygen will be enhanced twice. It will be double the capacity of the original individual. So these two copies will be passed on to the offsprings. Therefore, the offsprings will be better suited to even those environments where the oxygen concentration is less. For example, in hilly areas. So if they migrate there, they will be able to survive in this changed environment. Now look at another aspect, that after duplication, let us say that there are mutations in one of the copy, original being performing its function and second evolving to interact with the original, that is alpha and beta. They are interacting with each other to add cooperativity, as in allosteric cooperativity, wherein the binding of the first substrate enhances or cooperates or promotes the binding of the next, even at the lower concentration increase. Small change in concentration will give you higher activity. So this is what we call allosteric or hyperbolic or allosteric sigmoidal curve. So in this case, when the subunits, they are performing the same function, that is they are binding the oxygen. So alpha and beta. In hemoglobin we have 2 alpha and 2 beta. First binding of the oxygen helps in binding the next one. So at slight increase in concentration, this gives you sigmoidal. So this changes the behavior of the oxygen binding from hyperbolic to allosteric, which can then further be regulated using either enhancers or inhibitors. So in this way, enzymes are evolving, that is using both point mutations than gene duplications, and gene duplication may enhance the cooperativity or may add new reactivity. So now let's have an example of adding a new reactivity in the synthase, that is OSBS synthase, which has minor promiscuous activity, epimerase. Let us check if duplicated OSBS gene with promiscuous epimerase activity can evolve into main enzyme with epimerase activity and add new enzyme activity for evolving the harboring individual with additional capability. Visit Uniprot knowledge base at uniprot.org and search for L-DL-glutamate epimerase. The search returns 6 sequences. Download these sequences. Copy these sequences, paste at the beginning of the already saved FASTA file with 80 sequences for the OSBS and save the new file with the new name such as 80 OSBS and 6 epimerase sequences.fasta with all files selected. Upload this saved file, that is 80 OSBS and 6 epimerase sequences.fasta at Promels 3D and submit for multiple sequence alignment. In the ensuing Promels 3D results, show the color alignment with the sequence order in the input form with the block size of 60 and consensus level at 0.8. It was clear from 3D structure in OSBS that conserved arginine at position 27 has several electrostatic interactions with the carbonyl groups of the main chain atoms of distantly placed serine 264, isoleucine 265, serine 46 and proline 47. For placing the catalytic residues in the correct 3D position to catalyze the synthase reaction of the OSBS. The arginine at this position has been replaced with alanine, serine, threonine, isoleucine or valine in these 6 epimerase sequences, indicating loss of the electrostatic interactions to fold the epimerase differently for positioning the same catalytic residues to catalyze the epimerase reaction. The presence of arginine in E. coli at position 159 reveals its electrostatic interactions with main chain carbonyl from isoleucine 286, connecting 2 regular secondary structures separated by more than 100 residues in the primary sequence. The replacement of this arginine by isoleucine or alanine in 3 epimerase enzymes again shows the difference in the folding pattern for positioning the same catalytic residues to catalyze new epimerase reaction. Replacement of glycine with alanine at position 288 reduced the catalytic efficiency by a factor of approximately 500. From 3D structure, it is clear that mutation from glycine to alanine at this position, that is at 288, results in the clash of the introduced methyl group in alanine with carboxyl moiety of the succinyl group in the substrate. Similarly, the replacement of glycine at this position with aspartate in each of the 6 enzymes is responsible to reduce main OSBS synthase activity and convert the main activity to promiscuous epimerase activity. We know OSBS is the main, that is the synthase is the main activity and epimerase is the promiscuous activity. During evolution, promiscuous epimerase activity was transformed into main epimerase activity. Therefore, this confirms the role of the mutations affecting folding pattern and substrate binding into the creation of new activities. We have seen that the OSBS is having 3 acidic residues which are coordinating with the magnesium ion, and the 4th position of the magnesium is coordinated by the carboxyl oxygen of the alpha carboxylate in the substrate. The residue 133 lysine will abstract the proton and will form the enolate anion. This will have an orbital with 2 electrons which will then form the double bond with the carboxyl carbon. The double bond in the carbonyl oxygen in the carboxylate group will give negative charge. This negative charge is then in the transition state is stabilized by the lysine 235 from the other side. Now, you see here, this is the main reaction, that is the abstraction of the proton and stabilization of the transition state. Let us say that in this transition state, the lysine 235 changes its 3-dimensional orientation slightly to come to the carbon of the carboxylate group. In this orientation, when the transition state is stabilized by magnesium only, this lysine having a proton with it, and then here a planar structure developed due to the formation of the double bond between the alpha carbon and the carboxylate carbon. This double bond then, if accepts a proton from this 235 lysine, then it means the proton which was abstracted from this side will be added on to this side on the same position. This is what is epimerization of the substrate. So, this is the minor activity and converted to main activity just by reorienting the position of the K235 from stabilizing the carboxylate group to coming to donate a hydrogen to the alpha carbon. Similarly, we have seen that the synthase is converted to epimerase. Now, epimerase shall not be able to allow to bind the original substrate of the synthase. Therefore, a second mutation is selected, wherein glycine is replaced by an aspartate This creates steric clash with the succinyl moiety of the original substrate. Therefore, the original substrate for the synthase activity is not able to be oriented for catalysis of the synthase reaction. In this way, the mutation at 235 lysine helps in conversion of the minor activity to the main epimerase, and second mutation at 288 from glycine to aspartate blocks the binding of the substrate, so that now this enzyme is purely an epimerase enzyme. So, this is what we call the evolution. By just changing the orientations of the residues, new activity is created. In this way, the further new activities are also added on to the existing activities by just reorienting the amino acids required for catalysis. Therefore, conversion of epimerase into main synthase could be achieved with a single mutation from aspartate to glycine at position 288, as aspartate at this position 288 will not be able to block the orientation of the substrate for synthase activity. But the catalytic efficiency will definitely be much low, as several other mutations from synthase to epimerase has also occurred. Dear students, in this module, we have seen the evolution of enzymes for improving the existing activity, for surviving under changing cellular environments. For example, point mutations in OSBS for catalyzing same chemical reaction in enteric bacteria in GI tract or lactic acid bacteria in fermentation. And this allows, the point mutations allow the selection of the fit test to function under these different or changed environments, from GI tract to lactic acid fermentation. Similarly, we have also seen gene duplication, as in the case of evolution of hemoglobin, and subsequent point mutations for optimizing the new activity. In this way, nature allows evolve new species to survive under different environments with enhanced capabilities, either through addition of new reactivities or through addition of subunit interactions for cooperativity, as in allosteric nature of the hemoglobin. I thank you all for visiting EPG Pathshala.